All right, class. Now we will talk about the countercurrent multiplier system. And before we start, let me explain what I have drawn, and then we will continue further. If you look here, this represents the proximal convoluted tubule. The proximal convoluted tubule then comes down into the medulla, and this is called the descending limb of the loop of Henle. And then obviously this will be the ascending limb. This part is the wider part of the ascending limb. And then this is our distal convoluted tubule. Okay. All right. We also see the cortex and the medulla, and we do understand that the nephron loop dips down into the medulla as a hairpin structure. And then we also see the gradient in the interstitial space. We do see that as we move down the medulla, as we move down this way, there is this increasing salt gradient, right? We move from, so let me change color if I can. So in the cortex, it's 300 milliosmoles. But then as you move down deeper into the medulla, the solute concentration gets higher and higher and higher and higher. We move from 300 all the way to 1200 milliosmoles. What else is here? If you look into the wider part of the ascending limb, there are two bad boys in purple. Do you see this? These are cells. These are cells that line the lines the ascending limb. And to begin with, I just want you to know that these cells, they actually pump out salt into the interstitial space. So I just call them bad boys. All right. Are we good? All right. Now the question that I'm going to answer is why do we have this salt gradient in the medulla? Why? How is this happening? Who is bringing salt here? Why is medulla salty? That's a question we are trying to answer. All right. So we will start with the wider part of the ascending limb where we see the bad boys. Now as we look here, these cells have these. Do you see the blue transporters that I have drawn? All right. This is a sodium-potassium chloride cotransport. What is this? This thing is called a sodium-potassium chloride cotransporter. What does that mean? That means it brings in one sodium, and as sodium comes in, potassium will also come in, and also chloride will come in. Okay. So and what is here? What type of fluid do we have in the ascending limb? We have filtrate here, right? So from the filtrate, these cells are taking in sodium, potassium, and chloride. Make sense? Now inside these cells we have sodium, potassium, chloride. Good. However, do you see, so this surface, I forgot to mention, this surface is the apical surface of the cell. And then this one is the basal surface of the cell. So now these cells have sodium, chloride, and potassium. What will happen to these ions? The pink ball that I have drawn, this pink blob, that's our favorite sodium-potassium pump. So this pump will push out 3 sodium, and how many potassium will go in? 2 potassium in, right? 3 sodium out, 2 potassium in. Do you agree? So thereby sodium is being actively pumped, and of course this requires ATP. By this process, sodium is being actively pumped from the filtrate into the interstitial space. Do you see that? Great. What will happen to the potassium that are inside these bad boy cells? Well, let me do it in a different color. Potassium will leak out through these leak channels. So potassium comes out also. They'll simply leak out into the interstitial space. How about chloride, Rupa? Well, there are special pumps for chloride as well. Let me pick a different color. I'll just do black. So there are special channels for chloride as well, and chloride will also come out. Okay. In this regard, I want you to remember that a lot of sodium and a lot of chloride are pumped out from the filtrate into the interstitial space by these cells, by these bad boys. However, only a little bit of potassium leaks out into the interstitial space. So what do we get essentially? I want you to understand that essentially we end up getting tons of chloride everywhere, right? Do you see? And we also end up getting lots of sodium. So it's dense, dude. And we also have some potassium. All these salts, now we understand how exactly the salts are being pumped into the interstitial space. And all these this salt makes the medulla salty. It is because of the presence of the salt that you see this gradient, salty gradient of the medulla. Does that make sense? All right. Okay. So the medulla is salty, right? Very, very salty. I'll just put, and then think of the filtrate. Now let's move on to the descending limb. So, do we have orange? I really want orange, but no orange. So let me pick, what color should I pick? I'll pick this brown color. Okay, so let's start here. Proximal convoluted tubule. And the proximal convoluted tubule, the filtrate is at 300 milliosmos, which is isotonic to the blood osmolality. The cortex is also at 300 milliosmos. This filtrate will come down the descending limb. And if you remember, the descending limb is permeable to water and water only. So there are these cool channels. There are these cool channels lining the descending limb that lets water move. Now tell me, the filtrate is coming down. Which way will water move? Think, where do you have higher osmolality? In the medulla, so much salt, right? In the medulla, sodium, potassium, chloride. It's super salty, very, very salty here. Very, very salty. And the solutes suck, we know that. So water will leave. Through these channels, through these channels, and water will come into the medulla, into the interstitial space of the medulla. Does that make sense? And as we are losing, and how much water will come out? Well, water will come out until here it becomes 500. Come out 700, come out 900, come out 1200. The idea is the filtrate that is here, that is moving down the descending limb, will keep losing water until it becomes isotonic with the interstitial space. Okay. And so therefore, what's happening to the filtrate? The filtrate is losing water to the interstitial space and of course getting more and more and more concentrated as it's moving down the descending limb. And at the very bottom, the filtrate is super concentrated at 1200 milliosmoles, which is equal to the concentration of the interstitial space. 1200 here, 1200 here, equal. All right. What happens next? So now this fluid, the filtrate is here, and the filtrate is super concentrated. We agree, right? We started at 300 and we end up at 1200 milliosmoles. So the filtrate is super concentrated at this point. Rian, do you mind closing the door, please? Sorry, class. So the filtrate will move back up. It's a concentrated thing, moving up the ascending limb. But then think, the filtrate now moves up the ascending limb. And as it moves up, the ascending limb is 100% waterproof. So it's 100% waterproof. It cannot lose water. It cannot lose water. But through this ascending limb, as the filtrate passes like this, there are these bad boys lining the ascending limb who will actively, meaning using energy, who will actively pump out salt into the interstitial space. Right? So as salt gets out of the filtrate into the interstitium now, what is happening to the osmolarity of the filtrate? Think about this. So as the filtrate moves up the ascending limb, and as these bad boys, these bad cells pump out salt into the interstitial space, the osmolality of the filtrate starts getting down. So it goes to 900, then to 700, then to 500, and then ultimately when it's at the DCT, it can be actually 100 to 200 milliosmoles. It's getting diluted back because you are losing salt, in the ascending limb and not water. That's why the osmolality of the filtrate is going less and less and less, and that helps to lower the osmolality of the filtrate in the ascending limb, and it can go down to as low as 100 to 200 milliosmolality by the time it reaches the distal convoluted tubule. You might have to watch this video a couple of times to understand the concepts, but in all honesty, I have drawn out the exact same thing that I do in a regular class. So you are not missing out anything here. Feel free to email me if you have any question on this countercurrent mechanism. And oh, last thing, why is this called countercurrent? Look, the filtrate is moving down in the descending limb and up in the ascending limb. So down in the descending limb, up in the ascending limb. Is that opposite direction? Yes, that's why it's called countercurrent mechanism. And we call it countercurrent multiplier mechanism. All right, I'll stop here and then start with a new video which will explain another concept called countercurrent exchange system, and that will show you where will this salt and water go and how the vasa recta actually helps to maintain this solute gradient of the nephron loop or of the medulla. Okay, thank you.